So in this video, we're going to go through some of the anatomy of a mitochondrion, right? So one of the things to remember is that we hypothesize, and there's lots of evidence for this theory that mitochondria are really ancient bacteria that a long time ago, there was a prokaryotic bacteria that had sort of oxidative magic. It had the ability to do unique oxidative chemistry with kind of fuel oxidation. And another, early prokaryote said, hey, you're pretty cool looking. I like what you can do. How about you come live inside of me and I'll protect you, and then you can make energy for me. And that's sort of how modern day eukaryotes are proposed to have arisen. So we're going to see this story with mitochondria that could do magic with oxidative chemistry. And again, oxidative chemistry meaning they take electrons that are high energy and put them through an electron transport chain to be able to make ATP. Now, the difference here is those electrons are high energy by way of their chemical composition, coming from NADH or FADH2. In our next learning unit, when we learn about photosynthesis in plants and chloroplasts, we'll say, hey, chloroplasts are another kind of ancient bacteria that didn't have oxidative magic. They had light generated magic where they still did electron transport, but their electrons became high energy, not by virtue of their chemical composition, but because sunlight actually excited them to a high energy state that allowed them to go through the electron transport chain, generate a proton gradient, and then do oxidative phosphorylation. So lots that we're going to see here about these ancient bacteria and how they have become these important organelles in modern day eukaryotes. Okay, so to start this out, let's talk about what the structure of a prokaryote is. Okay, so if we think about a prokaryote, and I'm going to be very simple here, we're going to talk about there being an outer layer, which we're going to call a cell wall, and then an inner layer. And we're going to call this inner layer, okay, our cell membrane. So a little bit of anatomy, okay? We're going to call this is going to be the inside of the cell. This is the outside of the cell, okay? And then this region in between, you put it here, this region in between is called the periplasmic space. Now, we're not going to do too much with, prokaryote anatomy, but sometimes when we're looking at things, we might be looking at a prokaryote to understand how some things happen, so we might see some of this sort of anatomy. But I'm going to draw a red box here, which we're going to see in a minute how it relates to another red box that I'm going to draw on a mitochondrion. So let's contrast this to what we see with eukaryotes, right? Eukaryotes containing organelles. So I'm going to draw a very simple eukaryote here, and there's lots of organelles that are inside eukaryotes, but I'm going to focus right now on the mitochondrion. And mitochondrion we're going to draw as a very simple representation, sort of looking like this. So big picture with that story that I just highlighted, remember this theory, we call it the endosymbiont theory, basically says that millions of years ago, there was a prokaryote that had this ability to do oxidative fuel metabolism. And another prokaryote said, hey, why don't you come live inside me, do that oxidative magic, and I'll protect you. So this is this endosymbiont theory. This symbiotic relationship formed between a prokaryote that could do oxidative magic and another prokaryote that could protect that other prokaryote if they kind of merged together. So I'm going to zoom in here so we can sort of see this. We're going to see this anatomy and these words kind of like put together on another part of my slide here, but I want to put these sort of pieces here together. So when we think about sort of some of this anatomy with a eukaryote, we're going to say that this part is really the outside of our eukaryotic cell. This space here we call, and I can't spell right now, we call the cytosol. Okay, so that's the cytosol. And if we look at the details of a mitochondrion, I'm going to draw this red box here. And again, we're going to contrast how this red box compares to the red box we just drew with our prokaryote. But identifying things here, we're going to put these in kind of bigger words in just a minute. This is the outer mitochondrial membrane. This is the inner mitochondrial membrane. Okay, and then we've got a couple of spaces here. We've got the inner membrane space, and then all the way on the inside, we've got the matrix. Okay, so one of the things that I'm going to highlight here is I'm going to draw a red box here. So between this ancient prokaryote and a eukaryote that really has an ancient prokaryote inside of it as a mitochondrion, I'm going to redraw this red box here because we're going to draw the parallels that we see between the red boxes between these two different things. So each of these red boxes has two black lines in it. Okay, so here's one black line, here's another black line, okay? And highlighting here that even though I'm drawing this as a black line here, this is a lipid bilayer. So it's really representing phospholipids that are present as this lipid bilayer. These always take so long to draw, right? So not going to do too much here other than to highlight. There's our lipid bilayer. So you're drawing it as a line, but remember, it's a lipid bilayer. So let's kind of contrast, and this allows us to make sure that we say this is to that. We sort of compare and contrast these. So let's start with our prokaryote sort of on the left side here. If we call this the inside of the prokaryotic cell, and this is the outside of the prokaryotic cell. This space in between is called the periplasmic space. And we can label these two lipid bilayers as the cell wall that's on the outside and then the cell membrane that's on the inside. So again, we're not going to see this too much. We're really not studying prokaryotes per se. We're really studying mitochondria that are organelles that were ancient bacteria that are now within eukaryotic cells. Okay? So most of the time is going to be spent on the anatomy that's here on the right. So let's make sure we sort of see these comparisons. The outer sort of membrane on a mitochondria is called the outer mitochondrial membrane. So outer mitochondrial membrane, which is a mouthful. So we just abbreviate it OMM. Okay, and then we've got the inner mitochondrial membrane, which we're going to call IMM, right? The space in between here is called the inner membrane space. Which we abbreviate IMS, okay? And then we can orient ourselves that the outside is really the cytosol. Now highlight, this is not the outside of the eukaryotic cell, it's the outside of the mitochondrion, which is the cytosol. And then we've got matrix. Okay, now some things to highlight here is I'm going to change colors here to make sure to highlight this, that the outer mitochondrial membrane is very porous, okay? And what that allows for is the cytosol and the inner membrane space to have approximately the same concentrations of most stuff. Okay, so metabolites, ions, things like that, they're relatively equal between the cytosol and the inner membrane space because stuff can pass freely. And this is different from what we see with the inner mitochondrial membrane. The inner mitochondrial membrane is highly impermeable. And what that means is we can build up, concentration gradients of species. We can build up ion gradients. We can build up charge gradients, okay? And those are going to be all really important key features that allow the mitochondria to do the beautiful biochemistry that it's allowed to do. Okay, so making sure to sort of orient ourselves that the cytosol is equivalent to the outside of ancient bacteria. The matrix is equivalent to the inside of ancient bacteria. And the magic really occurs in either of these because of this highly impermeable inner membrane, which is the inner mitochondrial membrane for mitochondria, the cell membrane for prokaryotes, and again, the cell wall is equivalent to the outer mitochondrial membrane. So what we're going to see as we sort of continue on here is everything that's embedded within this inner mitochondrial membrane and how that's able to do electron transport and oxidative phosphorylation. So sort of as a translation to what we see in class, right? Remember, we've built our classroom to really do all kinds of neat things, whether it's being able to do hemoglobin highway or thinking about stage 1 and stage 2 of glycolysis, whether we're thinking about the citric acid cycle, and now moving into mitochondrial activity. We've built with our lab benches, right? That outer mitochondrial membrane, inner membrane space is sort of the channel between them, and then we'll have the inner mitochondrial membrane, and then all the way on the inside of that is that matrix. So we've got matrix, inner membrane space, cytosol, and we'll talk about what we're going to be building up in each of those compartments.